Hello and welcome to some biology class. Friends, today we shall discuss about cyclic electron transport. Photosystems 1 and 2 in the thylakoid membrane are not physically linked as implied by the jet scheme. Instead, they are segregated into different regions of the thylakoid membrane. One consequence of this heterogeneous distribution of photosystem is that photosystem 1 can transfer electrons independently of photosystem 2 in a cyclic process called as cyclic electron transport. In this process, the excited electrons, after leaving the photosystem, are transferred through a series of carriers and finally comes back to the photosystem. In terrestrial plants, the major pathway of photosystem 1 cyclic electron transport is thought to occur via P700 to an iron sulfur protein called ferredoxin. Ferredoxin then transfer electrons back to plastoquinone through a recently discovered protein called PGR5. From plastoquinone, electrons are cycled back to photosystem 1 by passing through cytochrome B6F complex and plastocyanin. In this process, the electrons are not used to reduce NADP plus into NADPH. Instead, the electrons are transferred through a series of carriers and finally comes back to the photosystem. Hence, the name is cyclic electron transport. In this process, there is no photolysis of water, so in cyclic electron transport, there is no release of molecular oxygen, and also NADP plus is not reduced to NADPH. As these electrons also passes from plastoquinone to cytochrome complex, cyclic electron transport also contribute to the establishment of the proton gradient, which drive the synthesis of ATP in accordance with chemiosmotic hypothesis. For each pair of electron passing from plastoquinone through Reyes iron sulfur center and cytochrome F to plastocyanin, 4 protons are translocated from stroma into the thylakoid lumen. Due to this proton movement, lumen become more acidic, whereas the stroma become more alkaline. The resulting proton gradient is then used to drive the synthesis of ATP in accordance with chemiosmotic hypothesis. Synthesis of ATP in association with cyclic electron transport is known as cyclic photophosphorylation. It is generally agreed that 3 protons must be transported through ATP synthase in order to drive the synthesis of each molecule of ATP. According to Peter Mitchell, the total energy available for the synthesis of ATP is called as proton motive force. Proton motive force is the sum of the transmembrane electrical gradient plus the proton gradient. Most of the evidences suggest that the steady state electrical potential in chloroplast is relatively small. So, most of the proton motive force are derived from the proton gradient. It is thought that cyclic photophosphorylation is the source of ATP for chloroplast activities over and above that required in the carbon reduction cycle. So friends, this is all about cyclic electron transport. For more updates, please subscribe my YouTube channel, Some Biology Class.